Hello, I'm Peter Midford, and I will be presenting today's webinar on comparative tools, searches, smart tables, and regulatory overview in BioCyc. Ingrid Kistler, one of our curators, will be covering questions in chat during the presentation, and I will cover remaining questions at the end. So today I will be covering a variety of topics, many of which deal with working with multiple organisms. This seems to be of interest as I've received several chat questions last week on working with multiple databases. So today I'll be looking at comparative genome browser, orthologs, blast searching, cross-organism search, multi-organism search, comparative analysis tables, using smart tables, and the regulatory overview. So let's start with the comparative genome browser. So the way you get to the comparative genome browser is on a gene page. There is a menu on the right side that Peter Karp mentioned last week, and you click on Align in Multi-Genome Browser. Select allowed organisms is basically a command that lets you decide what organisms you want to compare the current gene and the current organism to. This only works when the organisms you're interested in working with have orthologs. We've built ortholog links for. We have orthologs for many of our 19,000 databases, but not all of them. And in cases where we don't have ortholog information, we can make sort of a best guess, but the information will be displayed as not being orthologs. Display is centered and aligned on the orthologs in the other PGDB. So this is one of the reasons why having organisms with computed orthologs is important. And likewise, the coloring is indicating the presence of orthologs. So here's an example of the comparative genome display, and we've looked at here. We're looking at a gene called BETA, and as you can see, it's hashed, and the colors though indicate not operons as they do in the regular genome browser, but operons. I'm sorry, but orthologs. I'm sorry, they're not operons or orthologs. So let me show you an example. We'll start with one of our favorite E. coli genes, TRPA. Go to the general search, select TRPA, and now we are now we're in the genome browser. And as I said, we can go and show orthologs in multiple databases. And what this brings up is a chooser window, which basically lets us select a set of organisms. Now, if you've already set this up, then you may already have a set of organisms selected. However, I do not, so I will go ahead and select a series of organisms. Basically, all I'm doing right now, this is a chooser, sort of like the choose organism browser. So I'll choose B. subtilis, which is sort of an outgroup, and then I'll choose a couple of E. coli strains. Oops, I didn't want all those. Okay, let's see, salmonella. Someone has, looks like someone has not muted themselves. There we go. Search for that. Notice that I didn't actually have to put the species name. I could just go right to the strain identifier, and it picked that right up. And let's see, and whether we should go. Okay, it's not the one I was looking for. Oh, there we are. Okay, so now we have a set of comparison organisms, and then it takes us right to the compare it. Actually, this looks like it's showing us the Ortholog display. So let me go back and let's see, line in multi-genome browser. So here's the multi-genome browser. The colors in the browser, as I mentioned, are not indicating operons, although you can see some backgrounds that do show shared operons. The colors are showing orthologs. And likewise, there's hashing for the starting gene. And likewise, like the regular genome browser, you can zoom in. And as you zoom in, you see some more detail. And I would like to point out that if you notice for both E. coli and B. subtilis, it includes some regulatory information. We have row-determined terminator. We have some sigma factors here. And likewise, we have some sigma factors for B. subtilis and some terminators. But we don't have as much information for the other ones. And that basically just reflects the fact that E. coli K12 and B. subtilis 168 are curated, have been curated for a long period of time. And so there's a lot more information in those genomes than there are in genomes that we've, such as the tier 3 that we've just generated from RefSeq data. So we'll go back to the no, it's not going back there. Okay, I'll go back to Fravet then. Okay, are you seeing the which display are you seeing?
The correct one.
Okay, great. Okay, so then the next thing is Ortholog displays. We do have displays specifically for orthologs. As I mentioned before, not all organisms have computed orthologs. And we, again, start from a gene page and we can show orthologs in multiple organisms. Here and back to our gene page, and if we now say show orthologs, what this basically is going to show us is the orthologs for trip A across different organisms. It identifies the gene and the gene product, and it also has an operon diagram which shows where the ortholog is in relation to the operon that includes it for that particular organism. So for example, here we see in CFT 073, this operon seems to be missing a trip C gene. And then here's an operon where it looks like it's transcribed in the opposite direction for the enterica, for Salmonella enterica. Okay, we also have several varieties of blast search available, and these are done from the main menu. They're not done from the gene page. We can search for protein or nucleotide sequences. We can search for either a single organism database or all the databases in BioCyc. Searching, doing blast searches on single organism is very fast. If you know what organism you're looking for, that's the recommended way to go. If you search across all of BioCyc, it typically can take 5 or 6 minutes, and you really should just leave the browser window open for a bit, and it will return your results. And as I'll show you, the blast results page has links to the specific results, because the blast matches will return proteins and genes, and our results page includes links to those entities in our databases. So if I go up here to the tools menu, we see we have blast search current organism and blast search allcyc. Blast allcyc pretty much has everything that the single organism does, except it doesn't let you specify an organism. Let's go to blast search current organism. So what I'm going to do now is, even though we're looking at E. coli, I'm actually going to search for an ortholog in B. subtilis. It's going to be a protein. I'm going to use blast P, and I'm going to get a FASTA expression that I previously had generated. So here's my FASTA string. Go and paste it in. There are a number of other blast parameters that we can that you can modify if your search doesn't give you the results you want. It's search, and now we're doing the blast A search. It comes back, as I said, pretty quickly, and this is pretty much a standard blast result page, except that notice that we now have links to protein sequences in or protein objects in the B. subtilis database that we asked for. It includes scores and e-values, and if you can scroll through, and it shows the details of the matches as the usual blast output, again with links to the appropriate protein object. Actually, in this case, it's the gene and the protein object. Okay, I also mentioned that we have a sequence pattern search tool. And this tool basically lets you enter expressions in a sort of something like a regular expression pattern matching language. This is for matching short sequences in a very expressive manner, but generally this is good for searching sequences of 20 letters or less, whether it's proteins or amino acids, or proteins or nucleotides. Okay, another search that we have is a cross-organism search. Okay, I talked about pattern matching search. So cross-organism search is a way to do a text search for substrings in 2 fields. So every most BioCyc objects have both names and frequently synonyms, as well as what's a summary field, which is basically a description of the object as created by our curators. Now, tier 3 won't have a lot of information in summary fields, but most of our highly curated databases will have extensive strings in summary fields that this search will look for. Unlike the next search I'll show you, this has no limit on the number of organisms you can search for. It will search through the whole BioCyc database and give you matches across organisms you may have never heard of. You can also search against multiple types of objects, whereas the multiple database search is you basically have to specify which kind of object you're looking for. And we can match against either all or any terms in the set of terms that you provide for the search. So we go to cross-organism search. So what I'm going to do is I'm going to search for, I'm actually going to go ahead and type this in again. Okay, and then what I'm going to do is search for all terms in both names and the summary. And you notice it gives me a list of things that I could be types of things to search for. So I can restrict my search to proteins or enzymatic reactions, or I can just leave them as searching for everything. And then we do have the option of choosing organisms. We can choose lots of organisms, or we can't actually choose every organism in BioCyc, but we can choose a larger set of organisms. But I'll go ahead and submit this. And now what we get is a result table where we basically have, it indicates the common name, and then the organism, and then what the type of object is, and the sort of search relevance, which it sorts by this search relevance score. Now, not all of these names, of course, are going to have pyruvate kinase in them. Sometimes the phrase pyruvate kinase is going to show up in a description. And in other cases, it may show up as a synonym. So I can go back, and if I just do this with names only, and I still want to use names, so I can do it with names and any of the above terms. And I have my list of organisms already. And again, there are cases where pyruvate kinase appears as substrings in the name, and there are some others where it doesn't, but they may well turn up as synonyms. So for example, some of these enzymatic reactions. Yeah, most of these, okay, most of these look like they are actually showing up as react. Oh, here's one where you click on reactions. You'll see it has, this reactions page has a series of enzyme condition synonyms, and in fact, we'll find pyruvate kinase. Here we go, pyruvate kinase is in that substring. Okay, let's go and have a look at the multi-organism search next. This actually is a special case of the regular search. So if we go ahead and click on search for genes or proteins, so the multi-organism search is actually this sort of tan-colored box at the top of the search options. If we click on it, it all of a sudden gives us a select organisms. And as before, it's basically has the list of 6 that I entered before. You can change it here as well if you go to select. And so let's see, I'm going to search for, yeah, I think I'll go ahead and just try searching for gene trip A. And let's see what our query comes back with. Okay, it came back with the gene trip A across each of these across each of the organisms. That's what we'd expect. We can also try a substring search. So let's try D. I'm going to go ahead and submit. Okay, it did not like that. Oh, that's right. This is the gene name. Okay, let's try with a protein search. Oh, actually, no. Now, what we should do first is go to search for compounds. Keep those, and then here we'll use D-alanine. Okay, and now what we see for the D-alanine search is a lot of compounds across the different databases that have D-alanine as a component of their name. But again, this is limited in the number of databases you can select, and also it's focused on a particular type. So the D-alanine search didn't work when I was searching for genes. And again, as I said, for multi-organism searches, you can basically search for whatever you want. You need to click the search across multiple organisms box at the top. And this isn't a text search. You can actually search for any of the properties available for the type of object, whether it's a gene or a protein that you're searching for. Next thing is comparative analysis tables. This is a set of reports that you can generate comparing various features of the organism, of multiple organism databases. Again, it's this process of selecting a set of organism and a set of object types that you're looking at. Results a set of tables for each object type. Each organism is listed in a column, and there are lots of links for drilling down and looking at individual objects. And the report also includes a table of contents. Here it is over under analysis and comparative analysis. And now we have a page which lets us select both kinds of reports we want. So I'm going to select organisms and pathways. And if I select all, it does tend to be slow when you select all. So it's better to decide which ones you want to look at in particular. We have our current selections as before. So if I now submit this. Okay, so now we have a set of tables of results. I will point out that we do have a table of contents at the top to allow easy navigation. So we wanted to look at the pathway holes right away. We have a link for that. But what's in these tables? Okay, so the first thing is let's look at the database summary statistics. This database contains basically summary statistics for each of the organisms that we selected. Includes basically things like, genome size and number of chromosomes. It has a little bit of breakdown by genes, GC content. So there's lots and lots of comparative information from these databases. We can also, if you look at the links for the entries on the left column. Sometimes it's just shows this row, but in other cases, it might show a complete list of objects. So for example, let me find something fairly small. Pathways with experimental evidence. Actually, this is, so if you click on that, it will give you a summary table that shows pathways with experimental evidence. Here's the list in B subtle list and E. coli. There doesn't seem to be any experimental evidence in CFT 073. There's of course lots in K12. So that's the sort of summary table that you can see. Okay, there are other columns that have counts, and if you click on one of these count columns, it will again drill down to more detailed tables. So for example, if we look at REL 606, and we want to look at genes with experimental evidence, it basically shows us, here's the link for genes that have experimental evidence for their function and links to each of the genes pages. So okay, now for organisms, most of the other tables are simple. They basically just have values such as, so here's phenotype metadata. And so what we have is things like, are they free living or symbiotic, their relationship to oxygen. So all of these are basically just keywords that don't actually have pages. So it just shows them out as text. Likewise for collection, if we have collection information, for example, CFT 073 is a human pathogen. Enterica is a pathogen of several species, and Shigella is likewise for humans. We also have information about if we pull this genome from NCBI, what the RefSeq level is, whether it's a reference sequence or a or a typical or representative sequence, it will show up here. We go on to pathways. The first table breaks it out by pathway class. So as Peter Cart may have mentioned last week, we have ontologies, which are basically classifications of things like pathways. We have, in particular, we do have a classification of pathways here. So we have a class of biosynthetic pathways and then some subclasses below that. And again, we can go and look at a table of pathways in this class, and it will tell us if there are any, here's a list of pathways that BioCyc has added as classified as being in that class, and then the presence of that particular pathway. And if we click on this, it will go to the organism specific page for that pathway. And otherwise, we have totals, which would again take it to the list of pathways in that class for a particular organism. Okay, another table we have is pathways that are shared between pairs of organisms. So for example, how many pathways does REL 606 share with B. subtilis? Well, 141, and it gives a Jacquard similarity coefficient for these two sets of pathways. If you look down the diagonal, of course, all the Jacquard scores are 1. If you're wondering why the numbers here don't correspond to the numbers up here, so basically you'd expect every subtilis pathway is in B. subtilis, and so why is it not up to add it to 298? And the reason is simply that some pathways are represented in more than one class. Some pathways are included in more than one class, so that's why the additional 25 pathways in more than one class have showed up as this total here and not this total here. We also have a report of unique pathways. So these are, in particular these are pathways that are unique to the organism relative to the other organisms in the set. So because B. subtilis is our outgroup here, it has a relatively large number of unique pathways. It also doesn't hurt that it's well curated. E. coli K12 has a fair number of unique pathways, but in this case, it may be because it's better curated than some of the others. And we also have a report on pathway holes. Pathway holes are pathways that are predicted to exist, but where one or more genes of the pathway are missing. So in this case, we have total number of pathway holes and then fractions, and then we have links to pathways with one or more missing genes in them. So for example, if we look at pathways with 2 holes, we get a report for each organism, and these are the pathways in B. subtilis that although these pathways are predicted to exist, we don't have gene information for them. We don't have, they're missing one or more genes that we would predict would be there, and that's considered a pathway hole.
Peter,
Yes.
so there was a question from one of the attendees to explain again what the difference is between the multiple organism search and the cross-organism search.
Okay, the multiple organism search allows you to do basically all the search control features of a search in a single database. So you could search for all the protein, or all the compounds with a PI less than 4 across multiple databases. Whereas the cross-organism search is basically just a text search. It's looking for text strings in the names or the descriptions. So, I mean, I can go back and so basically if we do a multiple organism search, we say, okay, we want to do multiple organisms, and here we have gene, DNA, RNA search. So for example, we could search for, here we can do a search for sequence length. Whereas with the other search, the cross-organism search, all we can do is search by name. So that's the main distinction. Most people won't probably have large search sets of organisms greater than 70, but that's also another limitation of this particular search. Down here, or I should say up here, this list is limited to 70 organisms. So those are the main differences. One is a string search, the other is actually a semantic data search. Okay, smart tables. Smart tables are a very powerful tool within BioCyc. One of our curators, not Ingrid, has referred to them as sort of a bioinformatician in a box. What they let you do is, first of all, you create them by defining lists of database objects. So these are basically any kinds of objects that can be genes, metabolites, pathways, sequence regions. And they let you browse and work on their properties. You can transform them into related objects. You can also take these lists and filter and combine them, share them with everyone or with specific collaborators. And of course, you can export them to spreadsheets and in some cases, FASTA files and other sorts of external reports. They're also a jumping off point for omics analysis, but I won't really be talking about omics analysis this week. I will save that for Suzanne's presentation next week on omics. So there are 3 types of smart tables. There are smart tables that you create, and this is one of the reasons why you have to log in to work with smart tables. This isn't a subscription. This is the sign in as a user on the upper right corner of the page. So there are those that are created by you. There are those that are created by others, whether shared in public smart tables or or you can also someone can share a copy of smart table with you and you can make your own copy to edit. There's also the special smart tables. The special smart tables, basically, there's a set of special smart tables that include, basically, for each organism database, we have a smart table that has all the pathways, all the metabolites, all the genes. If you want to quickly get a list of all the genes in E. coli, this is probably the easiest way to do it. Go bring up this smart table and then save it to a CSV file, and then you can load it into Excel and you have all the genes. Other ways you can get to these smart tables include if you go to the summary statistics page, which is on the analysis portion of the main menu. There are a number of buttons for common entities in that summary page that you can also turn into smart tables. There's also, so smart tables are always associated with a particular database, and only objects in that database can be added to a smart table. Now, there are a few exceptions to this. You can have a smart table full of organism identifiers as well. Smart table rows and columns can contain PGDB objects and data fields. If when you're entering data into a smart table or loading it in from a file, if it doesn't recognize what it is, it will just say, okay, I don't recognize this. This is a data field, it's a string. And again, text strings can be entered that are not bound to a particular PGDB object. So if you wanted to create a smart table, you could type in metabolite names. You could, there are a number of pages where, as you may have seen as I went along, there are buttons that say, make this list into a smart table, and it will just go ahead, and if you're logged in, it will create a smart table with that list of results. You can also upload from a tab delimited text file. I'll point out, don't try to upload Excel files. Our text file processor does not read the compressed format that Excel uses. Use a CSV file with tabs. Actually, yeah, a tab delimited text file is what you need to use. And again, you can, oh, this is wrong, but okay, yeah, you can create a smart table by typing in names. And as I mentioned before, if you see this turned into a smart table, you can click on that and it will create your, it will create a smart table from the results. A number of things that you can do once you have this smart table is you can add additional columns that contain attributes. So for example, if you want the chromosome positions of all genes, you can also find the regulators of those genes. There are also transformations, which are more of a computational operation on the list of entities. You can create a smart table that contains transcription factors and then apply transformations. I'll show that in a moment. Other operations, because as I said, a smart table is basically a set of similar entities, is you can perform union, intersection, difference among two smart tables, and that will be operating on a list of objects in the left-hand column. You can also filter them to remove things or share them or freeze them. Freezing is a way to basically say, no one can modify the smart table anymore. It's going to be published, and we want it frozen as a URL that can be submitted in the publication. Okay, so again, to get to smart tables, we now have a whole column for smart tables, and I will go to, first I'll go to special smart tables, so you can see what we have available as. Okay, it looks like we're back in REL 606. Let me go back to K12. And before I go over to the smart table, notice, as I said, in the organism summary page, you can see shortcuts to creating smart tables. But let's go to the smart tables. We'll go to special smart tables. And here's our list of special smart tables. So as you see, we have all the compounds or all the genes, all the ribosomal proteins. We have a collection of these that basically contain all the entities of a particular type. We also have ones that have been shared with me. This is actually, it looks like me, but this is actually shared with me from another account. We have public smart tables, which are the tables that people have shared with everyone. So there are a number of active users that have shared various interesting smart tables based on a range of organisms for public use. I have the smart tables I've used before. So let's go back to the special smart tables and let's have a look at all genes of E. coli. And what this gives us is a smart table. As I said, the leftmost column, not the ones with check boxes, but this leftmost column is a set of gene names. And so what it has is, and actually each of these will go right to the gene page. We also have other columns that are properties of the gene. We have accession numbers, left end positions, right end positions, and the product. So now, what is the difference between properties and transforms? Well, they are in separate lists, and in general, a transform is an operation that involves more computation than, so for example, computing Go terms or finding the regulators of a gene is more computational, or is considered more computational than some of the other properties, such as retrieving an accession identifier or centrosome position or credits or data source. So these are, but basically the property columns and the transform columns pretty much work the same. So we can, for example, add a citations property for these genes. If we go down to citations, it basically adds a column of citations. These are PubMed IDs for citations that reference this particular gene. We could also, add a property regulated by. And again, it's added in a column. Once you have columns, one of the things there are a number of things you can do. You can sort by the column. So as you see here, we have sorts. So for example, the gene name here isn't actually sorted. So we could sort by gene name here and reverse the sort, or we could search by the accession numbers or these left and right positions. We can also change the name of the column by clicking on it. We could also, particularly for something like a product, we can actually, there's this little circle plus that gives us a set of operations specific. Okay, actually, it looks like it just jumped right to it, and it created a new smart table with the products of the different genes. So basically, one of the operations you can do on a column is take all the data from that column and build a new smart table from the set of entities in that column. Go back. So let's see, here are some transformations. We could go to, well, one thing we can do is look at the transcription units for a gene. And these are the names, these are the BioCyc identifiers of transcription units that include that gene. Transcription units are sort of include, but aren't limited to operons. But here we have some, here we have a transcription unit for the gene that we, in that row. So another thing we can do, let's have a look at a different set of entities. Let's look at compounds of E. coli. And when we open up the compounds, it gives us both the name as well as the structure of the compound. And again, we can add properties such as, say, the Gibbs free energy of formation. We have a database of computed Gibbs free energy of formation for for many of the compounds across our databases. We could also add a transformation of regulation genes regulated by compound. Yep. Okay, so there aren't any genes regulated by the compound. Let's see. Oh, here we go. Yeah, activator inhibited enzymes. That's the one I was looking for. So we can actually go and delete. Actually, what we can do, I think, probably would be better. Just collapse this column with the little left arrow. And now we can see that here are some enzymes that are inhibited by the compounds in this row. There aren't a lot, but there were a few up here where this enzyme seems to be inhibited by the presence of dihydroxyacetone. So let's go back to this operations menu. We can create a smart table. We create it as we can either create an empty one or upload a file, which again is going to be a tab delimited columns. We could load in some other files. We can type a series of data in, and basically what it will do is if we type in a series of identifiers, one line per entry, it will then try to look those up and tell us what it thinks each of those identifiers refers to. Another thing we can do is, of course, we can export our table. As I said, we can export to a spreadsheet file. If we have a set of gene sequences, we can export that to a FASTA file. And there are a number of other operations. Pathway collage is a nice visualization tool that Suzanne will discuss next week. Of course, we can delete the smart table or delete the column or set of rows. Again, we can do operations on columns and rows. So whenever you create a table, it's added to your list of tables. So if we go back to here to my smart tables, there are a couple that I've just created, and you can see from the update time that in the process of doing this, I've created 3 tables. But these tables aren't visible to everyone. So what you could do is, if we go back to this, if we wanted to share this table, we can click on share. And we could either make this publicly readable so everyone can read it, or we could specify a series of email addresses for people we want to be able to either read it or actually write it. This one isn't particularly interesting, so I probably won't. I mentioned frozen copies for publication. And so basically this will let you freeze a copy of the smart table so that it remains in a permanent form that can actually be published. Again, I won't actually do that. Okay, one last thing that I want to demonstrate for smart tables before we get onto the reg overview is enrichment. So what I'm going to do is I am going to new smart table, and I'm actually going to load from a text file. Let's see. This is a set of genes, and basically this is nothing special. It's basically just a list of E. coli gene identifiers that I pulled from a different data file. And I go ahead and say, yep, go ahead and make this object, and it has a set of genes. Now, the reason that I have just this random set of genes is that I want to show you how enrichment works in BioCyc. So because we have a set of genes, we can ask, does this set of genes unusually, what set of pathways are these genes represented in more often than by chance? Or we could do an enrichment for Go terms. But if we do an enrichment for pathways, it's then going to ask us, okay, which pathways include these genes among their reactions more often than you would expect by chance. So we'll say enrichment that we say more often than by chance. We'll use the Fisher exact test, and we will not use a multiple comparison correction. Go. And what we get a series of pathways, and in some cases we'll get sets of genes that are overrepresented in that pathway. Here, because we have a relatively small set, it only gives most of these are all single genes overrepresented, and we get the set of p-values without correction because we didn't ask for a correction. Okay, the regulatory overview is accessible via tools Genome Regulatory Overview. It basically displays transcriptional regulatory interactions among genes when the database contains them. So a few of our databases, about 10, have enough information that you can actually display a useful regulatory overview. E. coli, K12 being, of course, one of them. BioCyc databases can contain regulation for both protein and small RNA transcriptional regulators. And transcriptional information is also found in the Regulon tab of gene pages and transcription unit pages. The regulatory overview diagram basically has 3 sets of ovals. Each of these ovals is a set of genes. The ones in the middle are genes that regulate a lot of other genes. The ones in the middle regulate a fewer number of genes and are frequently regulated by ones in the middle. And the ones on the outer oval are genes that do not have any regulatory activity, but naturally are regulated by genes in the inner ovals. We have a right side operations menu, as we do for all of our overviews. We have a data layer box because you can plop a lot of data onto this regulatory overview, and this box gives us control over what is seen. We have opacity, which you'll also find in some of the other overviews. It basically makes these gray entities, the genes, sort of fade out relative to the diagram lines that are overlaid on top of them. And then we have zoom controls, and we can either use this zoom control or we can use a mouse wheel, which is our preferred way of doing it. Okay, so let's take a look at the regulatory overview. Tools, go over to Genome and regulatory overview. Actually, let me show you something first. Let me show you how to find how many regulations are in your database. So if we say by organism, so if we go to the organism chooser, select by organism properties, and select number of transcriptional regulatory interactions, and let's say we want, okay, is greater than 50. Okay, we want to add constraint and find organisms. And we can see that there are 11 that have more than 50 transcriptional regulatory interactions. These are the information that are being displayed on the regulatory overview. So if there aren't many here, then your regulatory overview is going to show a lot of information. So the inner circle has genes that, as I roll over, have lots of regulatees, which means they regulate lots of other genes. And we also have those in the middle where we have some there regulate fewer, but may also be regulated by ones in the middle. And then we have the ones out here, which are basically may have regulators, but don't regulate any other genes. As we roll over these, we can see details for the gene. We can hit, if we hit the keep button, it basically means the window stays around. You can move it around and it keeps tracking it. It of course has a link to the page for the gene. Let's look at how we can display actual regulation data. So if I click right on this, we can either highlight the data, we can write a report, or we can start highlighting, which is how those arrows that you saw in the diagram show up. So we could look at, highlight this gene and its regulatees, and it gives us regulation arrows to each of the genes that it regulates. We also have the overlay control lets us turn that one on and off. We can also change to include indirect regulators. And this will show a larger network of basically not just the genes that it regulates, but those that are regulated by those that it regulates. If I select a second gene, say if I select this gene and look at its regulators, they'll show up in a different color. And again, you can control whether those are displayed or by those checkboxes. Another thing, as I mentioned before, is you could go and save the network to a file, and the file looks something like this. So here's an RNA regulatory gene that basically has a list of the genes that are positively or negatively regulated by this particular RNA product. Another thing you can do is change the view. This is a little slow. So we can change it to top to bottom. And basically what this does is it says the ones that regulate a lot are in the top, the ones that regulate fewer are in the middle, and the non-regulatory are at the bottom. And we can switch that back. We can also display highlighted genes only. And this gives us the top to bottom diagram that basically shows just the genes that were involved in regulatory relations. So that's what I have, and I'm willing to take questions. I know I'm running a little over, but happy to take questions. Ingrid, are there questions?
I answered some of the Q&A questions. One of them you answered yourself as well. There was a question about how to match proteins from a data set in a particular species. I have no idea how to pronounce it. Comagatabacter europaeus.
Okay.
In particular, I think we might need to look at the data set and what identifiers there are in the data set and how to possibly get those matched. But people show up as Peter Midford in the chat. Oh. So I don't know who this was. So I just typed in there that please get in touch with Peter or to our support address via email afterwards if you need more help, because I don't know whether we can get in touch with you specifically directly from here.
Yeah, just go ahead and send that to BioCyc support, and we will resolve. We'll do what we can with that, because oftentimes information, we can map your information into our databases and do some sort of matching with it. And you did check the chat as well as the Q&A?
Yes.
Okay, great. Yeah, I have no idea why it's showing up as me there. It wasn't happening. I'm not sure what I did to that. But. So if there aren't any more questions, we will be meeting at the same time next week. I will be handling Q&A, and Suzanne Paley, who is one of our longtime developers and really knows just about everything there is to know about BioCyc, will be presenting on omics analysis. Okay, I think that's it. See you next week.